The second type of patterned secondary structure in proteins is the beta sheet. As pictured here, the backbone of these regions is mostly extended and hydrogen bonds form between adjacent backbone groups. These bonds may form between different segments of the same polypeptide backbone, that is intra-chain, or they may be interchain bonds between separate polypeptide backbones. There are two possible orientations of these extended regions. In a parallel beta sheet, as illustrated here, the hydrogen-bonded chains are oriented in the same direction with respect to their N and C terminals. The hydrogen bonds in our figure are represented by dashed lines. As you can see, the alignment is not exactly linear. In the case of antiparallel beta sheets, the hydrogen-bonded chains run in opposite directions with regard to the backbone. Note that in this arrangement, the hydrogen bonds adopt a more linear alignment. Can you imagine which of these two alignments might produce a more stable structure? If we look at a side view of a beta-pleated sheet, we get a better idea of how the structure gets its name. As you can see, it does not produce a flat, but rather a zigzag structure, like a pleat. Notice that the hydrogen bonds in this case are perpendicular to the backbone, not parallel as in the alpha helix. The side chains, pictured as the yellow spheres, alternate above and below the bonded structure. As we saw that there are irregularities in the structure of the alpha helix, so we also find irregularities in beta-pleated structures. What is known as a beta bulge can occur in antiparallel beta sheets, where a hydrogen bond involves two residues on one strand and one residue on the other. A classic beta bulge is illustrated on the far left, and other types of beta bulges are also shown. In the formation of the beta sheets within the same polypeptide chain, it's necessary for the backbone to fold back on itself in what is known as a reverse turn. An example is illustrated here, showing that these often contain the amino acid glycine. Recall that it is our smallest amino acid and can therefore more readily fit into the structure of a reverse turn. Another amino acid commonly encountered in reverse turns is proline. Its cyclic structure gives it the proper geometry to induce a reverse turn. As we proceed, we will see examples of protein structures that include alpha helices and/or beta sheets, and we can then more readily see how these structures relate to protein function.